Good evening once again. I would invite Deepa madam to start the session.
Good evening, everyone. So, am I audible? Hopefully, I am.
Yes, ma'am, you're audible.
Okay.
Yes.
So, my screen is visible, right?
Yes, ma'am, it is visible.
Okay. So, last class, we started with the Monera part and we were discussing about the bacteria parts of the Monera, right? So Monera, we learned about the basic characteristics of the Monera and we also started with the bacteria, the classifications and all. Hopefully you have all learned till there, okay? So what is Monera? Monera are basically the group of the prokaryotes. Unicellular, mycelial, colonial, and filamentous. And cell wall is made up of the peptidoglycan, polysaccharide, along with the cellulose, without any nucleus and the distinct membrane. DNA is naked, not associated with the histone proteins, and is the nuclear. This is just the revision part I am doing. So this is something we have learned the last class. So we have learned about the types of the Monera and also the types of the ribosomes it has and so on. Okay, so it has different kinds of the nutrition like the saprophobic, parasitic, chemoautotropic, photoautotropic, symbiotic, and some are capable of nitrogen fixation. They can occur in all environments we have learned, and we have learned about the kingdom includes prokaryotes like bacteria, actinomycetes, mycoplasma, and cyanobacteria. We were doing with the bacteria part. Okay, so bacteria, we have learned that it is the smallest free living organism which is mostly unicellular, and bacteria possess various forms and shape and are of different size like coccus, bacillus, vibrio, and spirilla. And we have also learned about this chart that we have learned about this chart in detail where we were so we learned. Yeah. So we learned about the different shapes of the bacteria like coccus, bacillus, vibrio, and spirillum. And we know that coccus is round, bacillus is rod shape, vibrio is comma, and spirillum is corkscrew shape. We all know this. Among them, I told you that diplo means two, vibrio means something which is in comma shape like the vibrio cholerae, and diplobacillus, then palisade bacillus, it's like a stack. Strepto means chain. Always remember strepto means chain. Staphylo means like bunch of grapes. Sarcine is 864 and so on. We have all learned this. Then we learned about the different shapes of the bacteria on the basis of their flagella. Flagella is something that helps in their locomotion like Iit-Jee, monotrichous, locotrichous, amphitrichous, peritrichous, and so on. So atrichous has no flagella. Monotrichous has a single flagella at one end. Locotrichous has single flagella at two ends. Amphitrichous has single flagella, groups of single flagella at one end, and peritrichous are flagellas all over your body. So this is something we have already discussed in the last class. So we also know that the cell wall is made up of the polysaccharide, proteins, lipids, peptidoglycans, murin. Murin and peptidoglycan, I told you, these are the synonyms. Both are the same. And polysaccharide, proteins, and lipids are the other functions we know. And plasma membrane is tripartite. Tripartite means three-layered. And mesosomes, these are the breathing, respiring organs for the bacteria. So this is something we have learned. So flagella is actually made up of flagellin. So pili is something which actually helps the bacteria during mating. Why? Because during mating, the pili actually transfers the genetic material from one end of the bacteria to the another bacteria. So this is the pili. Okay, so we learned about how to distinguish between two bacteria like gram staining We have learned this. So the gram staining is we heat smear the fixed stain of the bacteria, and then with the crystal violet we just put the crystal violet, and then we rinse with the water, adding the dilute iodine solution, and all the bacteria appears blue. Then we wash it with alcohol. Then the gram positive bacteria will have this purple color still even after washing with this alcohol, but the gram negative bacteria will be appearing red in color even after washing. Why? Because I told you gram positive bacteria has a teichoic acid and thick peptidoglycan layer, which the gram negative bacteria does not have. So because these layers are thick, they can retain the color, but gram negative bacteria cannot actually retain that color for that reason. So apart from this, we have learned about the episomes, nucleoid, ribosomes, granules, and so on. We have learned about the DNA of the bacteria. We have just learned that their DNA is just naked without any nuclear membrane. Okay, so DNA is actually what? DNA is actually a double helical structure which is actually circular in nature. It means that it is a rubber band. Rubber band, if it is in the twisted form, then it represents the DNA structure of the bacteria. Okay, so with these are the things we have already learned so far. And the incipient nucleus or primitive nucleus are named as nucleoid or genophore. So these are the things we have learned. We have learned about leader bug, and the plasmids having an independent existence, carrying genes for fertility, antibiotic resistance, which is the R factor, and I have already explained you the different types of the plasmid, like how even how the colchicine and the bacterium, these are being produced and how they are being transmitted and how they are helping in the survival of the bacteria. We have all learned about this in the last class. Okay, so till this we have done that obligate aerobes and the obligate anaerobes. I have told you facultative means something which is not very strict, but obligate means which are very much strict, like they won't be tolerating any kind of nuisance. If it's obligate means it will be doing that thing particularly. Okay. Acha. So now nutrition. So coming to nutrition, bacteria shows both autotrophic and the heterotrophic nutrition. That means autotrophic consists of the manufacturer of the organic material from inorganic raw material with the help of energy obtained from outside source. Chemosynthesis and photosynthesis. What does it mean is that bacteria can perform photosynthesis. Okay, remember this. Autotrophic means someone who has their own capability to produce their own food without being dependent on others. Like your mom is not autotrophic. Your mom cooks food for you, but she is not autotrophic because she is cooking the food that you are collecting from plants or vegetables, or sometimes she is even cooking the meat. And the meat is like the meat of, suppose the meat of a sheep or a meat of a goat, and that goat or a sheep is dependent on the plant for the survival. So we humans can't never be autotrophic. We have heterotrophic modes of nutrition. Plants are autotrophic. Plants are not dependent on food for any type of living organism. Plants perform photosynthesis. They prepare their own food, and with that own food, they actually circulate the whole body. That circulation actually helps the plant to survive, and the plant produce new fruits, vegetables, which are obtained by us. Similarly, bacteria is not dependent on any plant for photosynthesis or any human for any food. bacteria can show both autotrophic and heterotrophic. Like it can be doing its own photosynthesis and chemosynthesis, or it can be dependent on the other humans or other organisms for their food. So is this part clear why bacteria can be heterotrophic and why the bacteria can be autotrophic? What is the difference between autotrophic and heterotrophic and why humans can never be autotrophic? Is this part clear?
Yes, ma'am.
Okay, so since bacteria can perform their photosynthesis, obviously the chlorophyll of bacteria, there is a chlorophyll in bacteria also. Common sense, because plants perform photosynthesis with the help of chlorophyll. And when you will be growing up, you will know that apart from chlorophyll playing a major role in the process of photosynthesis, there are accessory pigments that actually helps the plant to perform the photosynthesis, to protect the plants from the extreme heat of the sunlight. Just imagine a plant is standing all the day long under sun, under the scorching heat of the sun. Are you going to give an umbrella to that plant so that the plant can survive? No, right? The plant has its own mechanism to protect its leaves, to protect its chlorophyll from the extreme heat of the sunlight, so that this chlorophyll does not get burnt, so that the plant can use this chlorophyll for the future reference. Okay. Similarly, bacteria have their own chlorophyll, and the chlorophyll of bacteria is the bacteriochlorophyll, and their accessory pigments are bacteriophyophytin, chlorobium, chlorophyll. So these two types of the pigments respectively occur in purple bacteria. So this name you have to again remember, like Theopedia rosy, Rhodopseudomonas, etc., which are present in the membranes of what? Thylakoids. Okay. So no oxygen is evolved in bacterial photosynthesis. Bacteria is not helping humans to survive, right? So if you know the process of photosynthesis, the light reaction and the dark reaction, you will actually know that in the light reaction of the photosynthesis, actually the plants evolve oxygen, right? The plants reduce water and produce that oxygen, so that the electrons are being promoted to the higher levels, and then they produce ATP and NADPH, and this ATP and NADPH are again being used in the dark phase or the dark cycle of the photosynthesis. So it's the light phase of the plants which actually produce oxygen. But do you think bacteria really need oxygen to survive? No. Bacteria can be anaerobic. We have learned bacteria can be anaerobic. They can survive without oxygen. But plants actually produce oxygen for their own growth, because plants don't have their oxygen. It's the oxygen of the plants that is extra, that is being used by us actually. Actually, plants need oxygen for their own growth. Okay. But the oxygen that is being produced, it's not like a property that plant can just make a deal that, okay, I am making this oxygen, I will be using it. Okay, so the oxygen that is being used by the plant is also used by humans and other animals. So that is the basic concept. But bacteria itself can live without oxygen. They are like very advanced type of organism. They can live with oxygen or without oxygen. So oxygen is not being produced by bacteria during bacterial photosynthesis, and such type of photosynthesis is anoxygenic photosynthesis. Anoxygenic means oxygen is not being produced. Water is not used as a reducing source, because obviously if water is being reduced, oxygen is being produced. So we are not bacteria are not producing oxygen. So why the water needs to be reduced, right? Instead, hydrogen is obtained either directly, like for purple bacteria, or from various types of inorganic and the organic compounds, like H2S, green bacteria, aliphatic compounds, purple non-sulfur bacteria. Okay, so but there are also bacteria. So is the bacterial photosynthesis part clear to you,
Yes,
everyone? Okay. So what are the chemoautotropic bacteria? They are like they can produce their organic food from their inorganic raw material. Okay. So with the help of energy, and where this energy comes from? This energy comes from these different kinds of reactions. Okay. Like nitrifying bacteria. Names you have to remember: Nitrosomonas nitrococcus, Nitrosococcus. They oxidize ammonia, and they produce nitrite. And that is the reason what happens is that energy is produced. Okay, so this is one of the classic example. So nitrocystis, nitrobacter oxidize nitrite to nitrate. Nitrite is NO2 minus, and oxygen, oxidization means oxygen is reformed and nitrate is produced. See, nitrifying bacteria are both Nitrosomonas nitrococcus, Nitrocystis, Nitrobacter. Both are nitrifying bacteria. But Nitrosomonas nitrococcus, they are just oxidizing ammonia to nitrite, and Nitrocystis nitrobacter, they are oxidizing nitrite to nitrate. This is the nitrogen cycle actually. Okay. Okay. So what is this? This is the sulfur oxidizing bacteria, Begiota. It is a colorless sulfur bacterium. This they oxidize what? Hydrogen sulfide, and they get energy. Thiobacillus, thiooxidants, they just oxidize sulfur in presence of water. Remember, one is oxidizing hydrogen sulfide, another is directly oxidizing sulfur in presence of water, producing H2SO4 and energy. So just during the NEET or any kind of exams, just see what they are asking. If they are asking you about only Begiota, then just consider H2S. If they are considering both thiobacillus, thiooxidants, then write this kind of reaction. Okay. What is the iron bacteria? Iron bacteria are ferrobacillus ferrooxidants. Reaction is again important. They are obtaining energy by reducing any ferrous, oxidizing any ferrous compound. You can see every whatever they are performing, they are producing energy by this reaction. And why this energy is needed? See, this energy is actually needed to convert inorganic raw material into their organic food. So they are not performing photosynthesis, they are doing certain reactions which produces energy, and this energy converts the inorganic raw material to the organic raw material. That is why they need this energy very much. Okay, so apart from this, methanosomonas oxidizes methane into carbon dioxide and energy. These are important, what I will say, important reactions that you have to remember. Next comes the heterotrophic bacteria. Heterotrophic bacteria may be what? Saprophyte, parasite, and even can be symbiotic. Saprophyte means suppose an animal has died, okay, or a leaf has fallen, or there is an animal excreta. Okay, so they are this bacteria leaves. So saprophytes are these organisms which are already dead. Okay, but what is the difference between saprophytic and the parasitic? Parasitics are not dead. Parasitic, just consider an example where have you ever heard of the term head lice? Hopefully, it is a very common term, head lice. Head lice is a parasite. It is not a bacteria, it's an animal, but it's a parasite. It does not kill the host. Rather, it slowly feeds on them. Okay. Saprophytes are someone who is already dead, feeding on those. Okay. And what is a carnivore? Carnivore means there is a living organism. The carnivore will kill that living organism for food. A lion will never eat a deer which has been dead for many days. Okay. So can I Could I make you understand the difference between a carnivore, saprophyte, and the parasite? The basic difference between a carnivore, saprophyte, and the parasite. Is this part clear? Okay. So symbiotic bacteria means they are in mutual relationship with each other, each and different kinds of the organism. Like Escherichia coli, live as a symbiont in human intestine. Okay, better to say Escherichia coli is a opportunistic pathogen. Why? Because Escherichia coli, unless and until it is residing in your intestine, it's fine. It will help you and promote your digestion. But when this Escherichia coli, for some reason, travels into your urinary bladder, then it causes infection there, which we commonly call as UTI. So E. coli is an opportunistic pathogen. Okay, so Escherichia coli. So maybe you have heard the term. Some people just can't take antibiotics for antibiotics. Why? Because antibiotics kill the Escherichia coli. Uh, I mean to say, some people can't take Escherichia coli and then they suffer from loose motion. Why? Because when this antibiotic is entering the body, this antibiotic is not specific. It is killing each and every microbe. Okay. So suppose for a fever, a doctor has prescribed the person an antibiotic, but that antibiotic is not so specific, means it can kill any kind of organism. That is the reason it is killing the fever wala bacteria and also killing the Escherichia coli wala bacteria in the small intestine. So after the antibiotic course, what happens? In intestine, there is no Escherichia coli. So obviously digestion will be hampered and this person will be suffering from loose motion. Not everyone is being affected, but maybe some person or the other is totally affected by this antibiotic course. So it's needed to complete the antibiotic course totally. Is it part clear? Is this part clear? So the parasitic bacteria live in contact with the other living beings, obtaining nourishment or special organic compounds required for growth. Bacteria has the four major phases of the growth in a fresh nutrient medium, like lag, log, stationary, and the decline. Lag phase is just growing. Logarithmic phase is exponential phase, stationary phase, and the decline phase. Okay. And there are three methods of the reproduction: vegetative, asexual, and the sexual mode of the reproduction. Okay. So before this, lag phase means the bacteria is preparing itself to grow. Log phase means bacteria has started to grow. Stationary phase means the bacteria has reached its maximum. So it's not growing because bacteria is growing in a medium. This medium has already been exhausted and the growth of the bacteria is maximum. So it has reached a stationary phase. Now the waste material of bacteria is accumulating nutrient on which it is being feeding on, it's being reduced, and that is why the bacteria is now facing a decline phase. So that's the lag, log, stationary, and the decline phase. Okay, so what is the vegetative mode of the reproduction? Vegetative mode is budding and the binary fission. So binary fission means during favorable conditions. So bacteria cells divide by a constriction into two halves, and this nuclear material also gets divided into two equal halves. So that is the simple binary fission. Asexual reproduction means endospore, conidia, zoospore. Endospore means unfavorable condition. So the bacteria forms a spore-like structure. That is the endospore. And bacteria reside within this spore. So endospore is a characteristic structure having the outer thin exosporium followed by one or many layers of spore coat, followed by concentric layers of cortex, which is followed by cell wall, cell membrane, matrix. Okay, so within this structure, this bacteria is present. So exosporium above exposed exosporium is the spore coat. So like this. Exosporium, then above exosporium, this is the spore coat. Then this is the concentric layer of the cortex. Then there is the cell wall. Then there is the cell membrane. And then there is the matrix of the bacteria. Okay, so this is the most external structure. This is the most internal structure. So this is the exosporium, spore coat. This is the spore coat. This is the exosporium. This is the concentric layer of the cortex, cell wall, cell membrane. This is the matrix of the bacteria. So these are the many protective layers that actually protects this bacteria from the harmful, unfavorable condition. So when the condition again becomes favorable, then these parts cancel out, and from the cell wall, the bacteria also comes out. Okay, because this bacteria has what? Normally a cell wall, then a cell membrane, then the cortex of the bacteria. We have learned this in the last class. Okay, so this part gets excluded and only the bacteria remains. This is in the favorable condition, and this entire structure is in the unfavorable condition. Okay, so endospore is highly resistant to very high and low temperatures, strong chemicals, acids, etc. due to calcium, dipicolinic acid and the peptidoglycan in the acid. It helps in stabilizing the protein. Whatever it is saying, it is that I have told you endospore forms when the bacteria is in very unfavorable condition, like in the strong chemical condition or a strong acidic condition or a strong temperature, very low temperature, very high temperature, whatever. So this bacteria forms endospore. It needs to protect itself. So DPA comes into function. So DPA actually helps in stabilizing these proteins. DPA calcium ions provide this resistance to heat. When favorable conditions come, an outer layer rupture, active bacterial cell comes out, and this is a method of perination. It means again coming back to original condition, and they say it is reproduction without multiplication. So what is reproduction without multiplication? Perination is actually the reproduction without multiplication, where they come back to the unfavorable condition, like where they overcome the unfavorable condition and come to the original condition. Okay. Okay. Sexual reproduction of the bacteria occurs through transformation, transduction, and the conjugation. Okay. So what is transformation? Transformation is simple. Two bacteria are present. Okay, bacteria 1 and bacteria 2. This genetic material of one bacteria gets converted into this bacteria. Sorry, gets transformed into this bacteria. Okay, so this is referred to as the transformation. And I have told you already how this happened because between these two bacteria, a pili-like structure forms. Okay, so this is the pili. This pili actually helps to convert, to transform this genetic material into this bacteria. So from this into this, the genetic material gets transformed. It happens only and only if non-capsulated to capsulated. If this bacteria gets capsulated, that means covered with a capsule like this process will not occur. Only if this bacteria, bacteria 1, from which the genetic material is being transferred, it is non-capsulated, then only this transformation will be successful. Okay? So just wait. So this was first studied by Griffith as in Diplococcus pneumoniae and is called the Griffith effect. So transduction is what? Transduction is actually where the genetic material of one bacteria like this. Okay. So understand this. So the genetic material of, sorry. So actually the genetic material of one bacterium goes to other bacterial cell by the agency of bacteriophages or phages. Transduction was first proposed in Salmonella by Liederberg and Zinder. So remember Liederberg did both the plasmid thing and the transduction thing. What happens? This is a bacteria and this is a virus. Okay. This is a virus. This virus will infect this bacteria. Done? And this genetic material gets inside this virus. Okay. So when this virus will infect another bacteria, genetic material of this bacteria which has now entered this virus will now be transformed into this bacteria. Okay, so this is the process for transduction. Is the process for transduction clear? This is a virus, this is bacteria 1 and this is bacteria 2. Okay, so this virus will infect this bacteria. This is step 1. The genetic material will get transformed into this virus. This is step 2. And this will be transformed into this. This is step 3. So these are the 3 steps for the sexual reproduction in the case for transduction. Okay. Now, transduction genetic material of the one bacteria will go into the another bacteria through bacteriophage. Bacteriophage is a virus. Remember, bacteriophage is a virus. What kind of virus it is? It is a virus that can actually infect bacteria. Bacteriophages infect bacteria. So it is a virus. So virus that infects bacteria, it is a bacteriophage. So viruses infecting bacteria are bacteriophages. Transduction was first reported in Salmonella by Zinder and Liederberg, and in abortive transduction, new gene does not integrate with the recipient genome and is lost. What it is being said, what is abortive transduction? Suppose this is the bacteriophage. Okay, this is the bacteria 1, this is bacteria 2. This is the virus, this is bacteria 1 and this is bacteria 2. And the genetic material of this bacteria will now be transformed into this. So in normal cases, this virus will infect this bacteria, step 1, number 1. Then this genetic material will go into this virus. This is step number 2. And then this genetic material will reach this. Then this virus will again infect this bacteria number 2. And then the genetic material will be transformed into bacteria number 2. But what happens sometimes, this genetic material cannot get transformed into the virus. This step 2 is hampered for different reasons. And abortive transduction is more common than transduction. Okay, so than a successful transduction. So if this does not get transformed, obviously, even if this virus infects this bacteria, this genetic material will not be transferred. Okay, so this is the abortive transduction. Is this part clear? What is transduction and what is the abortive transduction? Is this part clear? Anyone has any doubt?
Ma'am, can you repeat again what is abortive transduction?
Yeah, see. This what happens in normal cases. In normal case, this is a virus. Okay, this is a virus. I have written also it is a virus. So this virus will infect this bacteria. Okay. So the genetic material of this bacteria will go to this virus. Okay. Now this virus will infect this bacteria, and the genetic material of this virus will be transformed here. So actually the genetic material this virus has is this, right? So this genetic material of this bacteria gets transformed into B2 via virus. What is abortive? Here also the virus is infecting this bacteria, but the genetic material is not being inserted into the virus for many reasons. So now when this virus is infecting another bacteria, this genetic material is not getting transformed. So this is the abortive transduction. Okay. Is this part clear now?
Yes, ma'am.
Okay. And what is conjugation? Conjugation is simple. Conjugation is cell-to-cell union between two bacteria and the genetic material which goes through another through sex pili or through the conjugation tube. Okay, so remember, this transformation is from one bacteria to another bacterial cell by some unknown mechanism. Okay, but in what I will say in in conjugation, it has been established that the sex pili is present. Okay, so sex pili actually helps the bacteria to like this is one, this is another bacteria, sex pili forms, and then the genetic material gets transformed through this sex pili. So from the, just imagine it is a straw and the genetic material is flowing like this from one bacteria to another. So this is conjugation. Okay, so this is something you have to remember, role of bacteria in agriculture in increasing fertility. Azotobacter clostridium. Remember these names. These bacteria are nitrogen fixing bacteria. Okay, so plants need NPK. What? Plants need N, P, and what? K. Okay, for what? For their growth, obviously. Now, these plants will now, these plants don't have the ability to fix this nitrogen. So what they will do, they can't use the nitrogen obtained from nature. So they will ask for some bacteria to do it for the plant. So what is this bacteria doing? These bacteria reside in the roots of these plants like Azotobacter clostridium. And then they actually do what? They actually provide the fixed nitrogen to plants. So plant use this fixed nitrogen and actually grow because plant need NPK like you need complant to grow. Okay, so without that you are not growing. So you are getting it now, so you will be growing. So that is how plants. So that is how soil fertility is also increased by this Azotobacter and Clostridium. Symbiotic bacteria, rhizobium, fix atmospheric nitrogen, nitrosomonas, which converts ammonia into nitrites, which is further converted into nitrate by nitrobacter. We have already learned in that exam. In dairy industry, lactic acid bacteria, like Bacterium lacticeae and Bacterium acidilactice, convert lactose of milk into lactic acid and hence milk turns sour. Dairy industry lactic acid bacteria actually lactic acid forms. Actually, they form the lactic acid where and thus turns the milk sour. And this actually makes the paneer or curd and so on. Okay. In other industries, Azotobacter aceti converts sugar solution into acetic acid. Butyl alcohol and acetone are produced by the activity of Clostridium acetobutyricum. Rating or separation of fiber from stalks, examples in jute, sun hemp, linus, flax, etc., is done by water inhabiting bacteria Clostridium. Means for fibers in jute, they don't need water. So these bacteria actually separates the water. So that is this resin. Flavoring of the tobacco, like the cigarettes, the flavor, except the different flavors of the cigarette like mint, which are available now. We are not talking about that. We are talking about the regular cigarettes that we smoke, the original flavor. We can say that this cigars, I'm not taking the brand name, but this cigars taste better than some other cigarettes. So why? Because of this bacteria, Bacillus megatherium. Okay. Curing of the leaves of the tea is done by Myococcus condensans. Always remember these things because they come in the exams. Role of bacteria in sewage disposal. Bacteria and algae actually they decompose sewage. So they don't harm the environment. Plastics are actually can't be degraded, you all know, by bacteria or anything. So that is the reason plastics are being banned. Then what is the role of bacteria in petroleum? Petroleum pollution in water body is checked by Pseudomonas, because Pseudomonas is not growing unless and until clear water body is present. So obviously, if there is a petroleum pollution or not in the water body, that is being checked by the Pseudomonas. And what is the role of bacteria in human beings? Like E. coli, I told you that E. coli is present in intestine. It helps digestion. Sometimes antibiotics are being also produced by bacteria. Like we all know the discovery of penicillium, right? From penicillin. So that's an antibiotic. Then medicinally antibiotics are those organic secretions which destroy and check the growth of different pathogens without harming the host. Obviously, if we are using an antibiotic for a particular strain of streptococcus, then obviously that organism will die. So strepsil, we know the name of the medicine, strepsil. So actually streptococcus is an organism that causes the problems in throat when you are sick or having a cough or having a difficult time. So that is the reason streptococcus is actually playing an important role, and we are using antibiotic against streptococcus. Okay. See, this is very important. Subtiline Bacillus subtilis, polymyxin, Bacillus polymyxa, streptomycin and cycloheximide, Streptomyces griseus, chloromycetin, that is the chloramphenicol, that is Streptomyces venezuela. Terramycin, that is the oxytetracycline, that is S. Streptomyces rhamnosus. Aureomycin is Streptococcus aureofaciens. Erythromycin is Streptococcus erythereus. And neomycin is Streptococcus fradii. Okay, so these are the names you have to remember. Okay, spoilage of food happens how? Spoilage is the saprophytic bacteria growing, like on souring of milk, cheese, butter, their bacteria grows. Destruction of penicillin. So see, penicillin is also an antibiotic, but it is being destroyed by Bacillus brevis. Very important to remember. Cotton spoilage. It is done by Clostridium botulinum. Desulfurification of soil. Without sulfate, soil will not be healthy. So that is done, desulfurification of soil. That is done by desulfuricans. So these are the names. I know these names are all in the Latin form. We have to like, it's just a memory test and you have to remember this. Right. Reduction of soil fertility, denitrificans, Bacillus denitrificans, Micrococcus denitrificans, Thiobacillus denitrificans. They remove the nitrogen. See, azotobacter, nitrobacter, they all are fixing nitrogen, but these are actually reducing the soil fertility by denitrifying, means nitrogen is being reduced. So whatever the task, whatever the thing that is being done actually by the nitrifying bacteria. Okay, so one more question you can ask. So what is the benefit of the nitrifying bacteria with this? Imagine this is a plant. Okay, so imagine this is a plant and it is a leguminous plant. This is the soil, and here are the roots of the plant, and here the, what I will say, here azotobacter is growing. Okay, so azotobacters are growing here in the nodules of the root like this, and they are covering the roots of the plant and fixing the atmospheric nitrogen. So atmospheric nitrogen is being fixed by these organisms. Now, when this bacteria are living, okay, so this nitrogen is being produced by the plant and this plant is very happy. So this plant is very happy because this plant is actually growing happy. But what benefit is this azotobacter getting? So why does it helping the plant? We don't help anyone unless and until we get something in return. This is the law of nature. So what happens is that this azotobacter is getting shelter from the plant in the roots. Point number one. And sometimes this azotobacter also get nutrients. Like this plant is growing. This plant has some waste. It is the excess nutrient the plant is not taking from the soil. Just imagine, this is the soil where a plant is growing. Obviously, it means that the soil is fertile. So these excess nutrient are being used by this azotobacter for their own benefit. Now, again, think that for the plant, is nitrogen fixation enough? No. Just remember, when these nitrogens are being fixed, one particular organism is living. This will not allow any other kind of the organism to grow and survive. Suppose another type of the bacteria are being growing here. Like they want to live here in the same region. But these azotobacter are harmless, but these are harmful. Okay, so these are actually harmful. They are bad. So these are evil bacteria. So these are the bad bacteria. But these are the good ones. These are helping plants. So obviously, whenever the organism, one type of organism is living in a particular habitat, it will never want the other organism to survive. Azotobacter is not inhibiting this bacteria because they are harmful. Azotobacter will inhibit any kind of bacteria, even it is harmful or not, because this is the house for azotobacter now. And whether it is harmful or not, this azotobacter will always inhibit. Okay, so the plant is being protected even from other harmful pathogens just by giving them space. So is this part clear? So how the azotobacter is actually helping the plant? Because even if this bacteria are coming near them, this azotobacter will inform the plant and the plant will trigger its own immune response so that this harmful bacteria cannot attack this plant. Okay, so is this part clear? Whatever I have told just now.
Yes, ma'am.
Okay, so fine. So these are the things, which are the names of the disease. Very, very important slide because very frequently the questions come from this slide. Pneumonia, Diplococcus pneumoniae, typhoid, Salmonella typhosa, অথবা or Salmonella typhosa, or it can be Salmonella typhi, Typhae. Just note in your copy, it is Typhae, or it may be Salmonella typhosa. Typhosa I have given because typhosa is the latest term. Typhi was the old term. So, but in the examiner can give you the old name also. Vibrio cholerae, plague is Pastorella pestis, meningitis is Neisseria meningitidis, gonorrhea is Neisseria gonorrhoeae, syphilis is Treponema pallidum, diarrhea is Bacillus coli. Okay, what is this gonorrhea? Gonorrhea thing is a sexually transmitted disease. Gonorrhea, syphilis, both are sexually transmitted disease. In class 12, we have human reproduction and we will be learning about how to prevent these STDs. There we will be discussing these two diseases in extreme, not extreme detail, but in details. But just remember this, these are the gonorrhea and syphilis, these are the STDs. Okay, and diarrhea is the bacillus. Gastroenteritis is E. coli. Why gastroenteritis is E. coli? Because if E. coli reduce, if the population of E. coli reduces, then no one is there to help us, right? So that is the reason gastroenteritis is being caused by E. coli. Diphtheria is Corynebacterium diphtheria, tuberculosis, Mycobacterium tuberculosis, gangrene. Gangrene is Clostridium perfringens. Jaundice is Leptospira icterohemorrhage. Whooping cough is Haemophilus pertussis or Bordella pertussis. Tetanus is the lockjaw, that is Clostridium tetani. And bacterial dysentery is Shigella. Leprosy is Mycobacterium leprae. Okay, and one thing I want to say, for tetanus, this Clostridium tetani, this is an anaerobic bacteria. Okay, this is a facultative anaerobe. Means it will not tolerate any kind of oxygen. So this will grow only in the absence of oxygen. If there is a minute cut, for that tetanus injection is not required. But if that cut is deep, their oxygen is not reaching. So obviously, for a deep cut, tetanus is very much needed. Is this part clear? Is this part clear? Okay, so anthrax is Bacillus anthracis, black leg disease is Clostridium chauvi. Okay, so this is the entire chapter for no, not the entire chapter. There are other things. This is the entire thing for the bacteria. Okay. So any question you want to ask me? Tomorrow is a exam. So whatever we have learned so far, that will be included in tomorrow's exam. Okay. Is this part clear, everyone?
Yes, ma'am.
Anyone has any doubt, you can ask me. Hopefully no one has anything to ask me. So, sir or ma'am, I have completed the class. Yes, this topic is according to NCERT, strictly according to NCERT. Strictly according to NCERT. Okay. So anyone, I hope no one has any doubt. So whatever I am teaching you, it's basically just an extensions of NCERT because they don't give you from NCERTs, little bit extras from NCERT. So I'm just including and teaching you. Okay. Okay then.
Yes, ma'am. Thank you, ma'am. Yes, students. We have shared a Google form in the group for your details who have joined recently. So please fill the Google form. Then only you can get access to the YouTube classes. Okay? And also please share about the scholarship exam to your friends. Last and final phase will be conducted on 28th. So please share that information with your friends. Let them also write the exam. Okay. So again, we'll meet tomorrow morning from 5 to 7. Morning 5 to 7. Please join on time.
Ma'am, today physics class is not there.
No, we don't have physics class. Morning itself it is completed. You didn't attend morning class, Ifra?
No, ma'am, I didn't attend.
Okay. Morning itself it is completed. You have to attend morning classes. Morning we have chemistry and physics. Okay.
Ma'am, today's morning classes recorded videos are there?
Yes, they are there. You can
Okay,
just
ma'am.
go through the Zoom link which is shared today. In that you can just attempt. There you have a YouTube link. Okay.
Okay, ma'am.
So, okay, students. Bye. Again, we'll meet tomorrow morning 5 to 7.